Hey everyone, it's Mr. Sinti, and today I'd like to talk to you about another classic in the central dogma of molecular biology, which is the concept of translation. We know that DNA transcribes itself into RNA, but we're now going to talk about how RNA turns itself into protein, the second half of that central dogma. And basically, when you think about translation, you're talking about the conversion of a polynucleotide, the nucleic acids, come together in the cytoplasm and they basically turn you into flesh and blood. That's what we're talking about. We're talking about in this conversation how the genetic code can be translated into a polypeptide sequence and therefore a protein, which really manifests into all of the physical traits and all the enzymes and basically everything that the cell has to do with regards to protein. So let's just jump right into that. And so let me see if I can increase the size here. So I want to talk to you about this process right here, which is, there seems to be a lot going on in this diagram. And so what's going on, first off, is that translation occurs in the cytoplasm. And it's called translation for a reason, because you're translating a nucleic acid nitrogen base sequence into an amino acid sequence. So it's translating one polymer into another. And the genetic code is written here in the sequence of nitrogen bases of messenger RNA. Remember, in a 5' to 3' direction. And it's in units called codons and 3 nitrogen bases. And as you can see here, transfer RNA, which is sort of like this candle-looking structure, we'll talk more about it in a moment. It's sort of like a taxicab. In the cytoplasm, it picks up free amino acids and transfers them over to the ribosome. And so it's delivering the amino acids, and then the ribosome is going to be linking those amino acids together to form a polypeptide. And so what's kind of neat is that all three of the RNAs come together here. Transfer RNA is bringing amino acids, holding the protein in place. The rRNA, it composes the structure of the ribosome, and messenger RNA is the recipe itself. And so here's another close-up of that. And so here are the three types of RNA. There's transfer RNA, ribosomal RNA, and messenger RNA. And so we mentioned before that the codons are in groups of 3. And if you recall, Marshall Nuremberg determined the genetic code, or cracked the code. And so we can say that UUU is a code for phenylalanine. Whoops, go back to that. You may remember that it's phenylalanine, because curiously enough, that's what he used, the poly-U tail, to determine a little mini protein with phenylalanine. And so transfer RNA is a really cool molecule. It looks, you think of RNA, you think of single-stranded, but what you really should think about is the fact that RNA, because of its single-stranded, has the ability to fold back on itself. It's not really necessarily linked to its counterpart in DNA. It's only one strand, and so it can actually fold back and forming these interesting structures. There's a place right here where it attaches amino acids, and another place called the anticodon, where it corresponds to complementary codon bases. And so during translation, each type of transfer RNA will link to the messenger RNA carrying the appropriate amino acid. Let me talk a little bit more about this. Let me come over here and show you what I'm talking about. Look at this. This is a cool diagram right here. Not really a diagram, but it's really a animation, computer animation, showing the three-dimensional nature of transfer RNA. I remember when I first learned this, it was described as a clover leaf. I think things have come a little bit further in that conversation when you look at this sort of computer-generated three-dimensional model of transfer RNA. It's able to hold an amino acid up here, and then down below it's able to provide the anticodon, which attaches it to messenger RNA. And so let's go over here for a second. I'm going to show you something. This is a really neat video right here about what I'm talking about in terms of RNA. Like when you look at the difference between RNA and DNA, DNA is a double-stranded molecule, so it tends to not be able to form these interesting structures. Because RNA is single-stranded, it can sort of self-fold, and it can form double-stranded areas like this, and it can form all kinds of interesting structures like transfer RNA, for example. Not only can it form transfer RNA, we're going to see that in the large ribosomal subunit, it's so complex in its nature that it can actually catalyze the formation of the peptide bond. And so therefore, because of the fact that it has these intricate folding abilities, it's more than an informational molecule. It can act as a catalyst as well. And so let's go back over here to the notes if we can. Go there, and then perhaps there. So we're going to say that each transfer RNA is going to arrive at the ribosome, and it's going to carry its own amino acid, and it's able to bond to the messenger RNA with an anticodon. And so if I were to draw, for example, here's messenger RNA. If this is messenger RNA, you would know that if it's being read in this direction, it's 5'. Over here would be 3'. So in other words, if I had a codon, let's do this, G, C, A, the anticodon, in other words, the docking region right here would be complementary to that. This would be in a 3', 5' direction, and you might have guessed it, it would be C, G, and U. And that temporarily forms hydrogen bonds between the transfer RNA and the messenger RNA, and that's what holds it in place. Nice. Okay, and so like I was saying, if the codon is UUU, then the anticodon would be AAA. However, this is most important, the codon itself is in messenger RNA. So when you look at your decoder, the UUU would code for phenylalanine. You never want to confuse the anticodon. The messenger RNA contains the genetic code. And then it deposits it. And so this is a copy of the genetic code, you might remember. So this is messenger RNA. So if the code is UUU, it would be first base, second base, third base, and it would be phenylalanine. So every protein starts with AUG, which is the methionine start codon, and there's 3 stop codons. So there's 61 codons that are coding for amino acids, and 3 of them are for stop codons, and that means the translation is finished at that point. We'll talk more about that. So the transfer RNAs are designed to pick up amino acids in the cytosol. We'll look at the details of that in a moment. And then they bring the amino acid to the ribosome, so they transfer it. And then once they drop it off, then they can go out again. So they're sort of not necessarily recycled, but they could be reused, just like a taxi cab that sort of picks up a passenger and brings it to their destination and then goes out and gets another one. So they're kind of useful that way. This is a close-up looking at the hydrogen bonds, and it looks a little bit like a clover leaf when you draw it like this. Here's where it attaches the amino acid up here, and then here's the anticodon down here, which binds to the messenger RNA. Again, a more detailed look at it. So here's the 3-dimensional transfer RNA. This is sort of a cartoon drawing from our textbook. Sort of looks like a candle. This is where it attaches the amino acid. Here's the anticodon. So just checking, the codon for this would be, down here on messenger RNA would be U, U, and C. And so if you wanted to know what UUC was, you'd have to come back here and say UUC, and you're like, hey, phenylalanine. So that would be, phenylalanine would be the amino acid that transfer RNA was carrying. And so what's interesting about it is there's 61 codons, and so you might expect there to be 61 different transfer RNAs, but the actual number of transfer RNAs is 45, so that should tell you something. That must mean that transfer RNA is able to bond with more than one type of codon if there's less transfer RNAs, and so that is actually the case. And so transfer RNAs can bind to a couple of different codons, and As it turns out, that third base position, we have talked about this before in terms of the redundancy of the genetic code, it also kind of wobbles a little bit. Like, for example, if the wobble position U on the anticodon, say that's U, it can bond to adenine or guanine. So there's a little flexibility. It's almost as if you had two hydrogen bonds holding the transfer RNA to the messenger RNA, you're good to go. So a little bit of a little detail, but it's kind of curious that there's 45 transfer RNAs and not 61. And so here's another picture. We had seen this before, anticodon. Okay, and so this conversation right here is about how amino acids get attached to transfer RNA, and so this occurs in the cytoplasm. So each amino acid is joined to the correct transfer RNA by this special enzyme. It's a synthase, but it's called amino acyl transfer RNA synthase. So it's kind of a mouthful. Amino acyl tRNA synthase, and this is shown here in gray. So what happens, if I can discuss this, is there's an active site in the enzyme that grabs an amino acid, and then you might recognize this ATP. ATP gets hydrolyzed, and a pyrophosphate provides the energy. And so the pyrophosphate is, you remember this from DNA synthesis, the pyrophosphate jumps off, which forms AMP. So that pyrophosphate then gets cleaved. That's a bit of a detail over here. And so what happens is the energy is attached to the amino acid, which provides the energy to link the amino acid to the transfer RNA. So basically, long story short, is that it requires a little bit of ATP in order for the enzyme to connect the amino acid to the transfer RNA. And then once the transfer RNA leaves the synthase, it's good to go, and then the AMP is released. And so that enzyme is kind of crucial if you think about it. It goes out there and grabs amino acid and sticks it to the transfer RNA. And so, as it turns out, there's 20 different synthases for each of the 20 amino acids that are going to be attached to the transfer RNA. And so that helps to catalyze the bond between, if you remember that, if I can go back a previous slide and show you a little detail here, it attaches it to this region right here, the 3' end. And if you look more, let me go back a couple more slides, it always attaches it to that adenine right there. And so that's what happens in the cytoplasm. And so here's another picture. This is kind of cool. This is our from our textbook here. This is a computer model of the amino acyl tRNA synthase, which is going to attach the amino acid to that 3' area. And then here's the anticodon down below. And so energy is needed for that. So let's direct our discussion now to the ribosome, sort of the cooking island of making a protein. So the ribosome is composed, you may know this from a previous discussion, a small subunit which is composed of proteins wrapped by rRNA, and a large subunit, which is more proteins, larger, wrapped by rRNA. These are produced separately in the nucleus, and they leave the nuclear pores separate, but they come together when they meet up with the messenger RNA. And so since this is such a huge structure, it's a very, very large, complicated conglomeration of many proteins. You might expect it's going to have a tremendous three-dimensional structure, and so it has these crevices. It has what's called the exit site and the peptide growing site and the amino acid arriving site. It even has a tunnel right there where the protein that's being produced exits. There's an input that takes messenger RNA in. It's rather awesome, the ribosome. And so what I want to say about that is that after rRNA genes are transcribed into rRNA in the nucleus, you may recall our conversation before that there's many, many genes for rRNA, which is kind of curious. They're sort of repetitive and they're next to each other along multiple chromosomes in tandem. And we believe that that's the case because you need to be able to keep up with protein synthesis. You need a lot of ribosomes to do that. So it's it's more important to have messenger RNA than it is to make a messenger rRNA than a messenger RNA. And so what happens in the nucleolus is that rRNA and the proteins wrap together and they form the large and small subunits. And then those things exit the nuclear pores. Good thing for the nuclear pores because these are rather too large to be able to exit through a membrane on their own. And so just to give you some, not that this is testable, but it's just for curiosity, eukaryotic cells usually have large subunits of around 39 proteins. Can you imagine that? So though we think of ribosomes as being small and there's millions of them in the cell, like little pepper in an egg. An egg, that's a kind of a goofy analogy, but the small subunit has 24 proteins, and so what's interesting is the small subunit needs to attach to messenger RNA first, and so we'll get into the detail of that. I mentioned in a eukaryotic cell, what's fascinating is that there is a subtle difference between prokaryote and eukaryote. I would think what's more interesting is the fact that they're more similar than they're different, but thank goodness there's a little bit of a difference, because you may be familiar with the antibiotic tetracycline. It's like, why can you take this antibiotic and it kills bacteria but it does not kill us? Thank goodness. It's because it specifically goes after prokaryotic ribosomes and it sort of paralyzes their ability to produce proteins, and it doesn't affect eukaryotic ribosomes, and so that's a good deal. How do you know the difference? You can crack the cell open and extract the ribosomes and spin them down, and they have different densities. The prokaryotic ribosomes are a little bit less dense. And so, as I was mentioning before, the large subunit has these different regions in it: the exit site, the peptidyl RNA binding site, and the A site. So I always like to think of this as the place where the amino acids are arriving, and this is the place where the protein is growing. I find to understand these three sites, so A, P, and E, the best way to do that is look at an animation, because it's basically a process that requires animation in order to understand it. And so let's take a look at that if we can. Move that over here. And so let's look at this video here. And so one of the first things that happens in translation is, you can think of it as, just like in transcription, it's broken up in three phases, and one of them is initiation. And so let me back that up just for a second. Do you notice here it's 30S ribosomal subunit? So this is showing in bacteria. We know a lot about protein synthesis from our study of bacteria, but the first thing that happens in translation is that the small subunit binds to a region right up here, just upstream from the start codon. And so when the small subunit attaches, that allows the very first transfer RNA to bind, and it always carries the amino acid methionine. And so it forms hydrogen bonds, and once that small subunit and the transfer RNA are attached, then the large subunit can attach as well. And so once that attaches, this area right in here is going to be what we consider to be the P site, and so it opens up this particular codon. Do you notice here it's CCG? So if you had your decoder in front of you, like for example, I happen to have mine, so I can pull that out. So CCG, if I look over here, and what I'm looking at is messenger RNA, and so I'm like CCG, proline. So that must mean that proline is coming in next. Let's check it out. So when it arrives, it'll arrive here at the amino acid arrival site, and this is over here, the exit site. You're going to see why that's important in a moment. So the A site is expecting proline, and look, here it is arriving. Just pause it for a second. This is the part that I always find kind of weird. It's like, how does the transfer RNA know to arrive? I don't know that it knows to arrive. In my take on this, I think it happens so quickly it's hard to imagine, but all of these transfer RNAs, the 20 different ones are like bang, 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 bang. Actually, there's 45 different ones. Or maybe it was 41, I'm sorry about that. So they come in, they're like bang, bang, bang like this. And only the one that's possesses the correct complementary anticodon could bond. And so check that out. CCG codes for proline, but the anticodon is GGC. So this is the only one that gets to park. And so when it parks, temporarily, let's pause this here for a second, temporarily you've got two amino acids which are close together, and that's the whole point. You want to form the peptide bond. And so there it is. There's the peptide bond, which is catalyzed by the large ribosomal subunit. And then the whole ribosome moves over, it's translocated. And then notice here, this transfer RNA leaves the exit site. This is the area where the protein is going to be growing. So there's the next amino acid coming into the A site. There's going to be the formation of the peptide bond. Notice how the codon codes for the amino acid. There's the peptide bond. It's sliding over into the P site, and then this transfer RNA, you guessed it, will take off and exit. So this continues over and over and over again rather rapidly. And here's a new transfer RNA coming in. So here's how the code is literally translated into an amino acid polypeptide sequence. That continues on and on and on until you reach a stop codon. And when there's a stop codon, there's no corresponding transfer RNA there's 3 stop codons. And at that point, the protein is finished, and so it's completed. Not necessarily. When the protein is produced, it's the primary structure, and then it naturally forms the secondary and tertiary structures. But there's some post-translational modification that can occur. You can attach some functional groups, and it takes on some even more elaborate conformations, which is pretty cool. So let me close that down. Let me show you this. This is yet another video showing the process. This is a good one. And so here it is. Let's take a look at this. So this one's a little bit more modernized. And so there's messenger RNA leaving, snaking out of the nucleus through the pores, right there. And so it's looking to find the ribosomal subunit. And you guessed it, the first one to arrive is going to be the small subunit, although this is showing the whole ribosome right here making the protein. So it's giving you the big idea first, that here the protein is being produced, and it could be any protein based on the recipe. And then the protein is finished, and then it gets modified. So first thing that happens is the small subunit attaches, and then that methionine didn't show it, but the methionine attaches, and then the large subunit. And so here's the messenger RNA snaking into the ribosome, and then these goofy looking structures cruising around are the excited transfer RNAs, and it looks like they already have their amino acids attached to them. That's nice. And so they bring it in. They're going to be bringing it to the ribosome. So if we could look inside the ribosome, you would see that they're coming in really rapidly. And so they're temporarily bonding down here. So it's codon with anticodon. So it's codon with anticodon, and they just take places. They switch places. They come in really quickly. They're attaching, and you can see the protein being produced up above here. And not showing any detail, but the large subunit is catalyzing that peptide bond. So that's kind of neat. It's amazing that this kind of thing is happening, and then the protein is coming off the top of it right there. So that's kind of crazy. And so let's go back over here to our, oops, not that one. Let's go back to our class notes. And so what I want to say about this is, I've been mentioning this, that the large ribosomal subunit is actually catalyzing the formation of that peptide bond. And when that was discovered, that was a pretty big deal. It shows that RNA can be a catalyst, and it's not just protein. And we saw this earlier too in RNA processing, when the small nuclear RNA or snRNA acts as a catalyst in the spliceosome when it's editing RNA. So that's kind of neat. And so translation, we've been through this. So can be broken up into 3 stages: initiation, elongation, and termination. In other words, attached to the messenger RNA, uh, grow the polypeptide, and then finish at the stop codon. So let's look at that. There's a little bit more detail that I want to attach to this, but I don't want to confuse you. It's just a little bit of information that we know recently. In addition to the messenger RNAs, we have some additional proteins that are called so-called factors, which aid in translation. And not only factors that help things occur, but there's some energy that's needed in translation as well. And the energy that we're using is not ATP. You may have remembering that ATP was needed to pick up the amino acid in the cytoplasm, but this time we're going to hydrolyze GTP, which I find to be kind of curious. Why GTP? It might be left over evolutionarily from somewhere in the past, which I kind of think is kind of curious. And so let's look at initiation. Initiation is when the small subunit attaches to the messenger RNA, and then the start transfer RNA that carries methionine attaches. And so that's the first thing. A little bit of GTP is needed and some initiation factors, and then that will allow the large subunit to bind. And so there you have the translation initiation complex, not too unlike the transcription initiation complex that we were looking at earlier. And then elongation can be considered, even that can be broken up into a couple of steps. And so one of the first things is that it's going to require some elongation factors, again, some additional proteins that will assist in the process. And it will also require some GTP as well. Some energy is needed for this. And so smaller picture, I'm not sure if you can see this so closely, but basically it's what we were talking about. It's the new amino acid coming in, some GTP is needed. Let me show you a larger picture here. The GTP is needed in order for the transfer RNA to attach, and then more GTP is burned moving the ribosome over. And so what you got going here is codon recognition. So a little energy is needed, and then do you notice here translocation is sliding it over. So the three steps of elongation is codon recognition, peptide bond formation, which is catalyzed by the large subunit, and then translocation where it moves over. So peptide bonds are forming during elongation. That's like the heart and soul of elongation. And then again, it's kind of like a cycle because it repeats over again. When this transfer RNA takes off, it'll pick up a new amino acid in the cytoplasm, and then you're ready to receive another one. And so how did the whole thing end? How does it terminate? It terminates when you reach one of these stop codons. And so there's 3 stop codons, as you can see here. And the detail of that is that there's a special kind of protein, shown here in yellow, that comes in and it bonds to the stop codon. There's no transfer RNA for the stop codon, so this release factor comes in, and what does it do? It cleaves the protein away from the last transfer RNA. So you need something to let go. And so, do you see how this release factor binds to the stop codon, and then it hydrolyzes or cuts the bond between the polypeptide and the transfer RNA right over here. And since that's cut, it releases the free polypeptide. This transfer RNA goes on its way, and that breaks everybody apart so that the large and small subunits go their way. And so what's interesting, it's like, oh, they're leaving each other, oh my God, but this small unit is looking to attach itself again. It can start the process all over again, as you can see here. Here they are attaching, and it's interesting is we now know that many ribosomes could be attaching to one messenger RNA, and so it's just a matter of time. This one just got there, so a little bit longer still, and then it's finished. You might ask the question, it's fair, it's like, how do we know that? How do we know that there's this thing like this, it looks like a polyribosome structure? Because we actually have transmission electron micrographs of this. You can't see the proteins, but you can see how this is happening. It's amazing. Can't wait to see how our microscopes are going to change our understanding of things in the future, the more we can see. And so there's this idea of polyribosomes, which can attach onto a single messenger RNA and translate it in just a few minutes. And so the last thing I want to say is that I mentioned this before, is that when a polypeptide is produced from the ribosome, you're like, I guess it's finished. Well, because it falls into it's made in the primary structure, which is determined by the messenger RNA, but then the secondary and tertiary. But there's more to it. There's these chaperones. There's chaperone proteins that we believe attach onto the polypeptide and help it form into more novel shapes. And in addition to that, there's little sugars that can attach, and whoops, let me go back. There's sugars that can attach, and there's some phosphates that can attach in other functional groups. And just sort of a finale to this whole conversation is that I mentioned this before in class, and that organic chemists are now busy creating their own amino acids. When you think about it, why be limited by the 20 that nature provides? Let's create our own amino acids. Let's create an amino acid that has a ketone group, for example. And you can get these amino acids to form all kinds of novel structures, which means novel functionality. Who's to say? So, like, is that working? Is this something that is just science fiction? Yeah, no. We could actually add those synthetic amino acids to bacteria and then add RNA to them, and then these bacteria will start producing these proteins. And so it's only a matter of time until computers can be able to simulate what these proteins are capable of, and then we can refine their shape. Who's to know? So where does it lead? I'm not sure, but I'm hoping that we proceed in an ethical manner in the future, and I guess that always comes down to our society understanding biology enough to make these decisions. So I hope you enjoyed this conversation on protein synthesis and in particular translation. Thanks for watching.